Great. Well, I think that we'll get started now. Thank you all for joining us today. I'm Christopher Soroshek, and it's my great honor to introduce our speaker today, Dr. Silvia Formenti. Dr. Formenti is originally from Milan, where she also obtained her doctoral degree prior to pursuing fellowships in radiation oncology and hematology at the University of Southern California. Dr. Formenti initially joined the faculty at USC as an assistant and then associate professor of radiation oncology prior to moving to NYU and later Weill Cornell Medical College, where she is the Sandra and Edward Meyer Professor of Cancer Research and Chairman of Radiation Oncology. Dr. Formenti has received numerous awards recognizing her excellent contributions to the fields of radiation oncology, cancer biology, and immunotherapy, but ones that stood out to me in particular include the Gold Medal Award from the American Society for Radiation Oncology, the Stand Up to Cancer Laura Ziskin Prize in Translational Cancer Research, and especially the Jessica M. and Nathan Bibliovich Award for Excellence in Mentoring Women Faculty. We're so pleased that you could join us today, Dr. Formenti, and look forward to hearing about your research. Just before we start, I'd like to encourage the audience to visit the website listed at the bottom of the title slide to get updates on the Connect to Science series, including a listing of the upcoming speakers. Also, please, during the presentation, please post questions to the Q&A section at the bottom of the screen instead of the chat box. And with that, I'll turn it over to Dr. Formenti. Welcome.
Thank you, Christopher, for the kind invitation. I hope you can hear me well, and we'll get started. So these are my disclosures. I receive research grants from multiple investigator-initiated trials that sometimes industry supports, and honoraria for advisories. And I'm going to discuss the off-label use of ipilimumab, which was a gift of Bristol Myers Squibb for the trial in metastatic non-small cell lung cancer we conducted years ago. And here you can see the pictures of Cornell and our new building on the right, where on the 4th floor is the Department of Radiation Oncology. So the way we look at cancer has changed dramatically in the last 10, 15 years, whereby progress in immunology and cancer immunology particularly have refocused us into looking at cancer as both a tumor problem, a cancer problem, a cancer cell problem, but also an host interaction and an issue of the immune system of the cancer patient. And our immune system protects us, in fact, from transformed cells through a mechanism through both innate and adaptive immunity to recognize these transformed cells and eliminate them, and sometimes capable to establish an equilibrium whereby there is some mutated or transformed cells, but they are remaining in situ, like in ductal carcinoma in situ, and potentially in some low-grade tumors like low Gleason scores, prostate cancer. But the cancer patients we're dealing with in the clinic, and I'm a clinician, so I'm very much on the front of dealing with human beings, are those who have escaped the control of the immune system and gone through cancer progression, whereby a completely different microenvironment and systemic immune reaction to cancer has been established. So the result of escape is both a less immunogenic tumor and a more tumor immune tolerant host. So they're really two targets. So it becomes obvious from a clinical point of view to start asking, how do standards anti-cancer treatments interfere with this landscape? So if we start looking from a different angle, how does surgery, chemotherapy, radiotherapy, or monotherapy interact with this? So approximately 20 years ago, we introduced the concept that radiotherapy can convert the tumor into an in situ vaccine. And the idea is that when you apply focal radiotherapy to a tumor, you not only send signals, inflammatory signals, and they're a mix of pro-immunogenic and immunosuppressive signals within the tumor and its microenvironment, but also, they release through cell death, you release potential neoantigens, and eventually, at least in a subset of cases, you're able possibly to induce cross-priming and elicit a tumor-specific immune response, and not only contributes to the local control of radiotherapy, but also through the mechanism of immunological memory, conveys a capacity to reject or control micrometastasis away from the radiation field. And that effect has been reported in the radiation literature for many years as an abscopal effect, and we'll get back to it in a minute. So I'm going to the next 50 minutes or so, I'm going to go over some of these effects of radiation in the immune system. I'm going to talk about the synergy of focal radiotherapy with immune checkpoint blockade. And I'm going to maybe focus a little bit more on the mechanism of radiation-induced immunogenicity, both in terms of the adjuvanticity of radiation and the antigenicity that radiation brings about. And then over the course of my talk, I'll allude to something that is much more of interest for radiation oncologists, how we best use this tool that can, in fact, perturb the tumor and maybe stimulate the immune system in terms of maybe different sources of radiation, dose fractionation, and which site we should irradiate to trigger this effect. So this is a very simple experiment we recently repeated, but we haven't ever published, and it just shows the principle. This work done with Claire Van Puy box, whereby you grow an immune competent mouse, you grow a tumor on the side of the mouse, in this case is TSA, and murine mammary carcinoma. And then we use different dose and fractionation to perturb the tumor with radiotherapy. And in radiation biology, there is a way to convert the dose and fractionation to achieve biological equivalent dose with this BD formula. And here we show that, compared to the no treatment where the tumor grows very fast, if you don't give an ablative dose, so a dose that is not really capable to control most of the tumor, only 5 out of 15, you get less survival effect. So this is a model where the survival depends on the growth of the tumor, compared to, in fact, if you give ablative doses and 20 gray times 2 in this setting versus 10 gray times 6 times. So local control in this system influences survival. But I'm showing you this very simple experiment because the message is that when we try now to re-challenge all the survivors, whether they had undergone the ablative treatment or not, but they survived the first challenge, compared to naive mice that were not irradiated before, we not only see much better rejection of the tumor, but also we see an impact on survival. So some kind of memory has developed against this tumor that enabled the mouse to be vaccinated somehow and reject the new challenge. So with that in mind, how do we translate it to the clinic? So as I said before, this has been reported in the past as an abscopal effect, so based on the same principle, in a metastatic setting, which is when the abscopal effects were first defined. You treat one of the metastases, you would expect with an ablative dose the metastasis to go away, but not necessarily for the other tumor to go away. And in fact, in some rare cases, the other tumor also goes away, the other metastasis also goes away in a clinical setting. And these authors have reviewed very carefully the literature and were able to pin down 46 abscopal cases over 45 years, so clearly not a common event. And it's not a surprise, it's not a common event, because we now know from many lines of evidence that for the time tumors are established, particularly in the metastatic setting, there is a very robust series of networks that guarantee and assure the survival of the tumor, the immune suppression that is necessary for the tumor not to be rejected by the host. And it goes through the mechanism of immunoselection of more purely monogenic clones, the recruitment of immunosuppressive immune cells, and of course, the immune evasion, both at the level of the tumor and its microenvironment, but also there are products from the tumor that are put in circulation that also inhibit systemically anti-tumor immunity. So this is a nice review from Sharma and Padmani Sharma and Jim Allison, where they made the point that for these new drugs, immune checkpoint inhibitors or immune checkpoint blockade to function, you need to start with something. You need to have some form of immune infiltrating tumor. And it's much harder for this, the removal of these breaks of the immune system that could influence immune rejection. much harder for them to work in a setting of cold tumors versus a tumor that is already immune infiltrated with just help through immunotherapy. So we started thinking that maybe radiation could be that agent that helps converting a cold tumor in a hot tumor, or maybe synergizing in an intermediate setting of cold to hot tumor with immune checkpoint blockade. And it turns out that work from others in our lab have shown many lines of evidence that in fact, in this mechanism of many mechanisms of escape, radiation could play a good role. So whether it is the upregulation of immune checkpoint blockade ligands. So for instance, radiation is a powerful inducer both in myeloid cells and in tumor cells of PD-L1, and the opening up of certain receptor and KG2D induced by radiation. The interference with some important cytokines, and some kind of induction of interferon, and upregulation, correction by upregulation of HC class 1. And each of these steps, radiation could have an effect, and sometimes even a negative effect. So a mixed set of effects, a concept I'm going to come back to in a few minutes. So early on in our research in this field, we were fortunate enough to receive from Jim Allison as a gift is monoclonal antibody against the CTLA-4 for mice. And we had in the lab ongoing this 41 mouse model, which is a very common mammary carcinoma model, syngeneic in an immune competent mouse, where you grow the tumor on the periphery of the mouse. And it's pretty aggressive. It's like a triple negative breast cancer, as you can see, a cold tumor. And very rapidly, within 2 weeks, develop lung metastasis, and the mice die of progression of lung metastasis. And we show that when you combine radiotherapy with this anti-CTLA-4 molecule, you get an effect on the tumor, fewer metastases, start seeing some survivors among the mice. And most importantly, when you test lymphocytes from draining lymph nodes of the mice treated by the combination, you finally can show that they are capable to react, as demonstrated by the release of type 1 interferon with an epitope AH1 and hemagglutinin, but not in the other subsets. So very promising data on the synergy with anti-CTLA-4. And then this is work we did in collaboration with Mike Dustin. He had a 2-photon microscopy system where you could follow in vivo in the mouse the growth of the tumor and visualize the kinetic of infiltration of CD8 T cells, in this case in green. And we're able to show the radiation by itself is not really powerful in recruiting CD8 T cell to the tumor. Anti-CTLA-4 also is not, but once you combine the two, you get a massive and remarkable and sustained infiltrating the tumor. So all of this good, but I want to somehow bring back to reality this enthusiasm, because radiation also can mediate very immunosuppressive effects, whether it's induction of TGF-beta. And here I just showed you as an example the induction of adenosine. So adenosine, a key molecule in regulating the balance between ATP, so as part of damage expression in ATP is a mediator of cell death, and it's certainly an important molecule in inducing immunogenic cell death. So once you induce ATPs through cell killing and AD+, there is a balance that is regulated by adenosine as a counterbalance that actually has a very strong effect, inhibitory effect on a bunch of important mediators of immune response. So how does radiation plays into this balance? And one way to follow this pathway is to monitor CD38 and CD73. And this is work done by Eric Wannenberg, recently published in Cancer Immunology Research, where he focused really his PhD under the supervision of Sandra De Maria, who's been my partner in this research for now 20 years, in looking at what radiation does. And it turns out radiation upregulates the non-canonical pathway of production of adenosine. And this is shown in mammary carcinoma, but I'm going to show in a minute in human cell lines as well. And it does it in a kind of a dose-dependent fashion. So is certainly creating a break, very similarly to what it does in terms of activating TGF-beta. And then if you block CD73, you start enhancing the response to, in this case, use just a single dose of 20 gray. As you can see, both in terms of delaying the tumor growth and few mice surviving the challenge of this tumor. But it's particularly effective if you knock down CD73, and this is a doxycycline regulated system to conditionally knock down CD73, and it was tested with and without the combination of an anti-CD3 antibody. And what it turns out is that the curves are identical, so it's no additional effect, but certainly the knockdown is very powerful in extending the survival of the mice irradiated in this model. So how translatable this is? Well, it is. This is a cell line of a human mammary cell line, and you see the same effect, again, with the dose dependency. And then importantly, when we look at a cohort of patients who were treated with anti-TGF-beta and radiotherapy in a trial that was published in Clinical Cancer Research recently, a couple of years ago, we were able to show that, and this trial showed a little bit of an advantage in the women that received in metastatic breast cancer, received a higher dose of TGF-beta inhibitor during radiation of a single metastasis. They had a little bit better survival than the ones that got the lower dose, but we did not see any abscopal effect in that trial. And interestingly enough, you can see that the majority of the patients, whether they got the high dose or the low dose, full versus empty circles, they all had an induction of soluble CD73, suggesting that we were triggering a break, a different break, while we were blocking one checkpoint. And again, refocusing us on the necessity of addressing multiple blocks into immunogenicity of radiotherapy, probably by a multi-pronged approach that targets more than one checkpoint at a time to see a clinical success. So the idea is to shift the balance from immunosuppressive to pro-immunogenic signals of radiation if you decide to use radiation to convert the tumor into an in situ vaccine. And slowly and gradually, we're elucidating some of these mechanisms, whether interference with the negative effects, I listed some of them on the left, or potentiating the positive effects of radiotherapy, and which are on the right. But very likely that in the reality of the tumors we see in the clinic, you may need to address multiple steps in the established networks of resistance to immunotherapy. So I'm going to focus now on some of the work we have done to elucidate this mechanism of radiation-induced immunogenicity. And we were lucky to convince Claire Van Puy-Box to embrace what seemed at the beginning a really boring project, which was to test the difference in dose and fractionation when you give radiation. We had generated preliminary evidence years before that was kind of surprising, that a larger single dose seemed to be less effective, that from a biological equivalent, a less powerful total dose and inferior biological equivalent dose given in a fractionated fashion. But in combination with immune checkpoint blockade, in a paper published in Clinical Cancer Research, first author is Day1 and co-workers, and Sandra and I are the senior authors, we had created that preliminary data. Nobody really was interested in dissecting. It turns out that radiation oncologists are the ones who get the kick out of dose and fractionation. The rest of society is not particularly intrigued by it. But it turns out that Claire, who was inquisitive and comes from the radiation biology training, wanted to embrace this challenge. And she built a model, this time with two tumor DSA growing on both sides of the mouse. And as I said before, you always need anti-CTLA-4 to really trigger a powerful immune response. Use this model to detect a response in the other nodules and make it like a beacon of an abscopal effect, or an equivalent of an abscopal effect, just to facilitate the course of the experiments. And here you can see that if you don't give radiation, both grow. You give radiation and CTLA-4 by itself, it doesn't do much. You give the gray radiation to the one on the left of the mouse, you get clearly a response in field, but no abscopal response, no response in the other small tumor. When you give the combination with anti-CTLA-4, no change. It is only once you give the combination of a fractionated regimen with anti-CTLA-4 that you see a change. And this, at least in the field of radiotherapy, is difficult to explain. Why a larger and in theory more powerful dose doesn't work as well as a fractionated dose. What are the breaks? So the first thing that Claire did was in a collaboration with Norm Coleman, did many years ago, now a kind of a gene expression screen for what radiation was doing. And here you can see the difference in the single dose versus the fractionated dose in terms of gene expression, and then at a protein level with a virus as a positive control in terms of induction of interferon, and here are the genes that control interferon. And why were we so curious about interferon? Because more and more evidence was emerging that type 1 interferon being crucial for the recruitment in the mounting of an immune response preclinically of the recruitment of the subset of dendritic cells, the BAF3 dendritic cells, that seem to be the proficient ones for cross-presentation and tumor rejections. So what turns out to come from this experiment is that, in fact, the effect of radiation somehow converge with existing mechanisms that we have in place to protect us from DNA viruses and retroviruses. Because once you induce through radiation breaking of DNA, you have fragments of DNA that eventually, and whether it's high dose radiation or high LET radiation or low LET radiation, these fragments of DNA reach the cytoplasm, and in the cytoplasm are sensed by this machinery that is in place, in fact, to induce interferon to protect us from viral infections. And that is mediated by SIGAS STING, and I will not spend a lot of time on this pathway, but what is important is that is exactly the same pathway that we have in place for protecting us from infections. And Claire was able to show with antibodies against the sDNA that after 8 gray, you could identify this presence in the cytoplasm of cells of fragments of DNA. And then when you measure them, much to confirming what we saw in the in vivo experiments shown before, the single dose of 20-30 gray were at least associated with less detectable double-stranded DNA in the cytoplasm compared to the fractionated dose. And it seems to be a threshold dependent, because after 8 gray, you started seeing it, you lose the effect after 20-30 gray, and it's sustained more if you repeat it, and it's kind of nice to demonstrate that the repeated daily radiation dose of the correct dose and fractionation seem to be a powerful inducer of interferon. And here you can see through gene expression as well as the receptor expression and the protein expression. So the why is so important, as I said before, it turns out that the stimulatory effect of radiation, in fact, recruits BAF3 dendritic cells. And here you can see the number, the amount, and in terms of the dendritic cells, in terms of the different dose and fractionation in vivo. And also, most importantly, it activates them. And that could be demonstrated by the evidence of activation markers, CD40 and CD70. So are these essential for the effect of radiation? So we knock out the transcription factor BAF3, and in fact, we saw the abrogation of the abscopal effect and much less in-field effect as well. So this, at least in this model with a combination of anti-CTLA-4 and focal radiotherapy, BAF3 were indispensable, and we repeated in a couple of other models. Similarly, we remove the receptor for interferon, and again, we were able to show the wild type versus the knockout for the receptor with the abrogation of both the in-field effect as well as the abscopal effect. And then the necessity for having CD8 T cells were demonstrated by an antibody blocking CD8 that in yellow abrogated both the effect in the radiated side and the abscopal side. So we then ask the question of whether is the derived in the interferon really depends on the cancer cell. There is quite a bit of data showing from other groups, from Ralph Weichelsbaum and others, that the effect could be induced by the microenvironment, and in theory is not necessary to for the cancer cell to produce interferon. But at least in our model, it seems to be key that the cancer cell were in fact the source of interferon, and that was demonstrated by creating a doxycycline control system where we could conditionally abrogate whether SIGAS or STING expression. And here I'm showing you the specific results. I think I'm showing you, this is on cGAS, I think, yes. Where in fact, if we were able to block cGAS and the system will stop working, as you can see. So this is in the irradiated side. And this is once you give the CTLA-4 when it's not silenced, and you irradiate one side, and both in the abscopal side and the irradiated side, you see the effect. But once it's silenced, you lose the effect. So, and we repeat the same for STING. So this is all important and relevant because in some tumors, the cGAS STING can be methylated or can be lost, the function can be lost. So you need to have different mechanisms to elicit a type 1 interferon response when you give radiation. So what I've shown you so far is that in the mechanism of radiation mimics that of viruses in eliciting cGAS STING pathway that induce type 1 interferon that are crucial for recruitment of the BAF3 subset of dendritic cells, and radiation is also enabling the maturation of these cells as well as their effect in cross-priming in the presence of anti-CTLA-4 that elicits a CD8-mediated response that in fact results on an abscopal response outside the radiation field. So all of this is very much apparently, at least in our data, influenced by the dose and fractionation. And I'm going to show in a few minutes also something about the side dependence. So in the same set of experiments, Claire went after a potential mechanism for not seeing cytosolic DNA after single dose radiotherapy, and she identified at least as one of the mediators, TREX1. And here you can see the TREX1, which is is well known for, in fact, removing fragments of reverse transcribed HIV virus and blocking the interferon response in that setting. TREX1 is in fact induced after single dose radiotherapy, and you can see very drastically induced after 20 gray, but not after, and when the silence, of course, is not there, but not after, much less after 8 gray. And that is reflects on interferon expression. So if you knock down TREX1, you now recover the immunogenic effect of 20 gray of radiotherapy, and now you can detect the dsDNA in the cytoplasm. So a barrier that seems to be elicited by large dose of radiotherapy and the pathways governing this induction, we're still trying to work out, and it's not that simple, but we're in part of the lab of Sandra is looking at it, and Claire are looking at it. So, and then is this really relevant not only on the field effect, but also on the abscopal effect of this large dose of radiotherapy that before was not as successful? So when you look at the wild type, you really don't, if you knock down in the wild type setting without knocking down TREX1, you really fail to see an abscopal response. You start seeing it after you have knocked down TREX1, both in the irradiated as well as in the, and you see more when you repeat it twice, the 20 gray per fractions. So once again, a fractionation is better in red compared to just a single time with CTLA-4. So all of this nice in the mice, but we impatiently want to translate it to our patients. And one indirect evidence is that when you look at the clinic and the papers published, and this is a beautiful work done by a consortium of investigators who tried to test it, pembrolizumab in combination with single, site, just one site of stereotactic radiotherapy in advanced solid tumors. And they look at different dose and fractionation, somehow dictated by the constraints of normal tissue. And interestingly enough, most of the time they went into the range of dose where we would not expect the synergistic effect of radiation. And in fact, the clinic demonstrated that they had a 13% objective response rate, which is very much similar to what you will see in an unselected cohort of patients that had never seen pembro or immunotherapy before. So radiation didn't really seem to help in this setting. Conversely, this is a prospective randomized trial from the Netherlands Cancer Institute where they compared one arm received pembro and radiation versus the other arm received the pembro alone, and you can show that the addition of radiation made a difference in terms of the overall survival of the patients, doubling the overall survival, 36 versus 16 months, the median overall survival, the objective rate, sorry, and the median overall survival from 8 months to 16 months. And interestingly enough, most of the effects that you can see here occurred in the ones, so this is the arm of the experimental arm, most of the effect occur in the arm that was PD-L1 negative, suggesting the radiation may be able in certain cold tumors to elicit, synergize with pembro and elicit, somehow induce PD-L1, and multiple people have shown the evidence that radiation can do so, and in fact enable the response to anti-PD-L1 or anti-PD-1. So we embark into the difficult task of figuring out this optimal study design to prove this concept of a combined, so translate our mouse work to the clinic. And ideally, you will want a large prospective randomized trial where you're giving immunotherapy alone versus immunotherapy and radiotherapy. And we're now fortunate we have such a study that is getting toward completion. I think it just completed, where in a neoadjuvant setting of early non-small cell lung cancer, we have randomized to durvalumab alone versus durvalumab by radiation, and we have the pathologic response as evidence of how we have modified the tumor microenvironment by the combination. But to see an abscopal response, you need a setting, a metastatic setting, and it's complicated to have homogeneous groups, and certainly to run, it's very costly to run a large prospective randomized trial. So another approach is to use radiation and immunotherapy in a setting where historically there is evidence that immunotherapy does not work. And the opportunity came for anti-CTLA-4 because in non-small cell lung cancer, whether locally advanced or metastatic, there was quite a bit of initial enthusiasm that was tempered by the modest objective responses, and pretty much anti-CTLA-4 alone was shelved after these results. So we mounted a study where we were combining radiotherapy to one metastasis. And while we were writing the protocol, we treated off protocol with a special consent patient who insisted to be treated as per protocol and knew about the protocol, who had a refractory metastatic non-small cell lung cancer, yet progressed through 3 lines of chemotherapy and chest radiation with multiple metastases all over the body and multiple liver metastases, and was sent to us really for palliative care. But he insisted, he read our results, preclinical results in the waiting room from the publication of the Cancer Center, heard that we had a trial in preparation, and we obtained a specific permit from our IRB, and were able to obtain as a gift the drug in this specific case, where specific consent was made for this patient. So we treated 1 liver metastasis here with a very modest radiotherapy dose, or ablative dose of 6 Gray × 5, certainly not an ablative dose, and we pulse ipilimumab 3 mg/kg with a standard every 3 weeks for 4 cycles. So much to our surprise, the patient obtained a magnificent response very relatively early, within 2 to 3 months, with disappearance of at least the PET-CT of all the lesions in the liver, little bit of a persistence of some fullness in the hilum, and these are kind of almost disappearance of this pleural base lesion that he originally had, and the response in the bones as well. So this patient, however, at this moment in January, still had a palpable supraclavicular node, pretty much in the same position where he had been diagnosed, even if you can see that he was not picking up at PET-CT. He allowed us as a gift to remove the lymph node to study it, and was generously permitted the surgery for us to study the lymph node, and we were able to compare to the original supraclavicular node that made the diagnosis in this case in 2010. And in this case, we showed that after giving EP and radiation to the liver, there was a nice infiltrated increasing in CD8 T cells and a ratio of CD8 to FOXP3 that was favorable. And then we were able to do some sophisticated staining on these lymph nodes, and we show, in fact, PD-L1 upregulation. But instead of interpreting it as a sign of exhaustion, it looked actually as much more as a sign of activation, because 12% of the CD8 T cells were Ki67 and coincided with the PD-L1 expression. In fact, this patient never got PD-L1, was only treated with CTLA-4. He got another 2 cycles of ipilimumab, and then were left alone. And it's now 7.5 years later, NED with no other treatment whatsoever. So are these cases that keep us enthusiastic about testing this combination and continuing this line of the research. So based on these patients, as I said before, we had in preparation and confirmed by this patient, we already had in preparation a trial of combining ipilimumab and localized radiotherapy in chemorefractory metastatic non-small cell lung cancer. And we were testing, depending on the site we were treating, what was the safest, whether it was 6 gray times 5 or 9.5 gray times 5. And then the trial was designed, as I said, with the gift of ipilimumab from Bristol Myers Squibb, but was funded internally by the department, and we had multiple blood sampling, and then we used an NIH R01 to do the analysis of the blood sampling. There were 39 patients that accrued to the trial. Of course, there was a lot of skepticism in the cancer center about this study. And as you can see, there were many times at the end of their course, 41% with brain metastasis, with most of them with ECOG of at least 1 and more than 1, and with multiple lines of previous therapy. And in fact, only half of the patients were able to tolerate and complete the 4 cycles of ipilimumab. However, much to our surprise, for a kind of subset of patients very unlikely to respond to anything, and particularly to respond to ipilimumab, had already failed in earlier non-small cell lung cancer, we started seeing some CRs. And most importantly, we saw some interesting molecular correlates of the responses. And in general, with this 18% response rate, we kind of reposition the ipilimumab in metastatic non-small cell lung cancer because radiation could make it work much better. And then we did a random forest analysis of all the markers we could think about that could have been associated with response to give us lead in understanding why some patients responded and some didn't. And really, the tumor mutation burden didn't really pan out. There was the type of expanded and contracted clones seemed to make a difference, the pattern of expansion of clones, as well as the induction of type 1 interferon, and I'll show you some evidence. So the very important translation that we could confirm in this study, probably the most robust information, was, as we showed before in preclinically, the responders tended to all increase on the amount of type 1 interferon in the blood compared to baseline, and was early, was before the 2nd cycle of ipilimumab. So if radiation works with ipilimumab, is very early on in the course, and we didn't see that pattern in the no-responders. And then probably another important learning came from a patient who achieved a complete response. This is a 61-year-old man who presented with brain metastases. They were excised, they progressed after first-line chemotherapy, had a lot of disease, progression, and the hilum, and bilaterally, supraclavicular, and so on. And he had undergone all brain radiotherapy after the excision of the metastasis. And we just, they treated the hilum alone with 6 gray times 5, and he had a complete remission, a complete response of all sides, and is now alive on nivo 6 years later. So one thing we could do for the patients in this trial was to follow in the blood their tumor-derived clones. And there is a way, this is like a barcode of the CD3 CDR3 region sequence of the clones of T cells in the blood. And we could follow the change in the repertoire after the combination of radiotherapy and anti-CTLA-4. And then in this specific case where we had the tumor, we could in fact look at the overlap of the present infiltrated, the T cell present in the baseline tumor, as well as the peripherally expanded ones, because we had the metastases that were removed from the brain. And here you can see the course of these clones in this patient compared to other patients that either had a progression disease or stable disease. So the sequencing of the tumor enabled us to, in fact, start exploring for the presence of neoantigens that could be recognized by the circulating T cells of these patients. And we were able to identify 148 genes with somatic mutation that were expressed in 123, and somehow through the pipeline went down to 33 potential candidates. And then through the mechanism of stimulating the T cells of the patient with the peptide and measuring the interferon gamma production, we could go down to look for some specific ones. And in fact, we were able to identify a mutation in karyoferrin A2, which is a transporting. And the idea is that as part of the DNA damage response, the entire DNA repair machinery is evoked, and those are often genes with mutations in cancer. And if you, as stimulated by radiation, you could have the transcription and translation of some of those genes that are mutated, it could become neoepitopes. And this is the case in these patients where the peripheral lymphocytes recognize the mutated epitopes and not the wild type. And here you can see the clones occurring at the time of radiation and anti-CTLA-4 and maintained over time. So these are the specific mutations. And then what we did in a PDX from a tumor of another patient just showed in general that when you give radiation, you induce this karyoferrin, and in fact is a protein, is a gene induced by radiotherapy that makes a protein. So as a proof of principle, this is an example of radiation-induced neoantigen, an epitope from a mutated gene known to be upregulated by radiation in human cancer cells, both in vitro and in vivo. And this, together with induction of interferon, supported our hypothesis that radiation can convert the tumor into an in situ vaccine. So what I've shown you so far is this kind of fascinating viral mimicry of in situ vaccination, whereby radiation is capable to produce endogenous adjuvants, type 1 interferon through these DNA breaks that fragments are able to reach the cytoplasm and elicit Cgas sting and type 1 interferon. And there is a very nice recent paper from our group from the lab of Lorenzo Galluzzi by Taka Yamazaki, where the relevance of the mitochondrial DNA is brought to bear. So it is also important to understand the mechanism of radiation effects on mitochondria. And then at the same time that I just shown you, radiation as part of the DNA damage response, radiation elicits a bunch of mechanism of transcription and translations in the irradiated cells, and some mutated antigens can be made, mutated proteins can become epitopes for rejection. And in some patients, lucky enough to have a lot of matching with the MHC class 1 and all, a lot of things have to align, that could trigger a powerful immune response that contributes to their response to clinical outcomes. And here I'm showing you again the important effect of soluble interferon. So I just want to not to leave alone this very important lesson we learned. This idea that irradiating one metastasis is a home run to immunize the patient against the other metastasis is really unrealistic, and much of tumor biology that has emerged in the study of metastatic disease shows that, in fact, the metastases are very heterogeneous and can have their own life on their own, and can somehow metastatize from one metastasis to another site. So the idea is that this antigenic diversity requires a multi-site vaccination approach, and also different metastases can have different mechanism of evasion from the immune system. So that calls again for a multi-site approach. So if you decide to irradiate the liver, in the patient I showed you before, we're lucky the metastasis seem to really have a nice burden of shared epitopes with the irradiated liver metastasis. But in most patients, you may get an effect on some distant sites, but not in all of them because they're just different. And you have not elicited an immunization against them. So the idea is that if you want to use radiotherapy, it's probably a good idea to target all metastatic sites. And I've shown you some evidence that maybe there is a dose infractionation that works better than larger single dose or dose beyond a certain threshold. And some evidence of the value of irradiating all metastases coming out from non-immunotherapy. This is a classical radiotherapy trial conducted by our colleagues in Canada, Palma and co-workers. They put together a consortium with a Swiss institution and other Canadian institutions where they, in a small prospective randomized trial, they were able to compare metastatic patients treated with state-of-the-art and radiation at need versus upfront treated with radiotherapy to up to 5 metastases. And interestingly enough, with the exception of some of those patients, they were using dose infractionation in the range of what we would think you are in an immunogenic range. There was no immunotherapy in this trial, and just want to stress was a 2-to-1, 66-33 control kind of randomization, and 3 of the 66 had treatment-related deaths. So even with a cautious use of this dose infractionation, you can start seeing some toxicity. So one food for thought is that maybe we could reduce the dose of SBRT, be a little bit more cautious, and combine with immune checkpoint blockade and see whether in patients with oligometastasis we can see a difference. So I conclude by summarizing what I've shown you. The focal radiation has significant effects on the immune system, both immune suppressive and pro-immunogenic, with multiple opportunities for therapy. Radiation induces viral mimicry with cytosolic DNA accumulation leading to cGAS-STING pathway activation and interferon production with a dose fractionation effect. Radiation increases tumor antigenicity by enhancing the expression of immunogenic mutations. In combination with immune checkpoint blockade, radiation can convert the tumor into an in situ vaccine, and is now up to us clinicians to write enough clinical trial to really test how to optimize radiotherapy in this new application. And I want to recognize the multiple people that are working in this direction, certainly in our division of radiation immunity. Sandra, who's been my professional partner for now 20 years plus, and it's been a pleasure to work with. Laura Sant'Ambrogio, who just joined us a year ago, was an incredible asset. And Lorenzo, I invite all of you to read this beautiful paper in Nature Immunology, and as well as some new stars emerging. Claire Van Boy-Cox is not on this list yet, but Ari Marciscano has also joined our group. And of course, a lot of clinicians helping to make this happening, both at Cornell and NYU and Mount Sinai, the funding, and most importantly, our patients that make all these efforts worthwhile in the challenge that we are facing with them every day. Thank you very much.
Wonderful. Thank you so much, Sylvia, for that excellent talk. So just a quick reminder to the audience members, if you have a question for Dr. Fermenti, please just submit it in the Q&A tab at the bottom of the screen. And if I may, I'd love to ask a quick question. I was especially fascinated with your data on TREX-1 and was curious about what makes the TREX-1 only get induced at the higher doses of radiation therapy. What have you learned about the regulation of TREX-1 that could give you some insight into that? And I don't think I saw it, but I was curious as to whether the TREX-1 knockout cells also had an even improved abscopal effect at other doses of other dose fractions with the RT.
So we did not see an advantage in the normal, the doses we know they are effective, suggesting that the barrier is induced by higher doses. But I wish I could tell you more about the mechanism of TREX-1 induction, and I'm not capable, so I apologize. We're working on it. It seems to be more elusive than what we thought.
And are there some small molecule inhibitors potentially in development or available that could be tested clinically for this effect?
Yeah, so I know there are. One of the challenges we have encountered is that that threshold for TREX1 induction seems to be somehow model dependent. We saw it pretty consistently across 15, 20 models we looked, but there are mutations in the tumor that seems to change the threshold of TREX1 induction. So it may go down to lower kind of doses per fraction, 500, 600 in some specific tumors with specific mutations. So that is very intriguing because it opens an entire different area of research. And I'm not involved into that, but I know the lab is involved with that, yeah.
Great. Okay, so we have a few questions from our audience members. First one from Clemens Grasberger. He writes, regarding irradiated sites, is there a difference regarding induction of abscopal effect in bone and brain mets versus liver or lung mets?
So we used to think that this was the case, and mainly because Kwon and co-workers and the senior author, I think was Chuck Drake, had reported on an interesting large prospective randomized trial that Bristol Myers Squibb supported of ipilimumab versus plus radiation versus radiation alone in metastatic prostate cancer, where they were targeting bones and they didn't see a difference. As a note to that trial, I just want to stress that that trial that was reported as a negative trial in terms of objective response rate, it was a subset analysis, not planned subset analysis, showing that in fact, in the subset of patients without visceral metastasis, you start seeing a time to progression difference and a small trend toward overall survival improvement. But in that trial, it's really interesting because I recently saw the paper in the European literature showing a difference on survival that is coming out for the patients who receive ipilimumab radiation compared to radiation alone in that trial. So it started as a negative, but when you look at survival endpoint at 3 years, the ones who got the radiation with ipilimumab did better in terms of survival. So this is radiating bones. So I think before we say that bones are not a good source of epitope or not a good source for immunizing, I think we should be cautious. We have seen responses by subcutaneous lesions, by irradiating lung lesions, by irradiating brain lesions, by irradiating. So I think it has to do, in terms of detection of abscopal responses, it has to do with more with heterogeneity of the metastasis than the site you irradiate.
So that actually leads well into another question from Ping Pang asking, may I ask whether the combination has been tested in pancreatic cancer patients? If so, what is the clinical outcome? And perhaps you can just expand a little bit more on what other cancer types do you feel like these combination therapies may be particularly effective in?
So I know of preliminary unpublished data of efficacy of adding radiation in triple negative breast cancers. We are struggling, working very hard to try to combine radiotherapy with different forms of immune modulation in ER positive breast cancers. In the setting of pancreatic cancer, I don't think it has been tested enough. Is the pattern, one concern that I didn't elicit in my presentation, but I think has to be kept in mind, is that no matter how you give the radiation, there's going to be a blood flow going into, when the beam is on, being exposed to radiotherapy. And you want to make sure you do not induce lymphopenia, because it makes no sense to try to build a vaccine and at the same time induce lymphopenia. And circulating lymphocytes are extremely sensitive to radiation. So, particularly if you use with intensely modulated radiotherapy, which is a technical, physical way to deliver the dose from different angles, you may expose quite a bit of blood to a toxic effect of radiation that will somehow remove the warriors toward the war you're trying to build. So it doesn't make a lot of sense. So one challenge will be, when you treat multiple metastases, how do you monitor the circulating T cells and whether you have enough naive memory T cells that can build a really sustained response. In pancreas, there are metastatic settings to the lung and to the bones, but often there are local metastases, and it's likely that by treating those, you will also treat a lot of blood. Now, there are ways around. You can start thinking about IL-15. You can do other things to promote an expansion of T cells that you may need during the vaccination.
Great, thanks. So there's another question for Tomas Radiovic. Is there an optimal DNA fragment size for interferon induction, and does it match with the 8 gray?
So thank you for that question. So we have an ongoing tension in the lab, because I'm very intrigued by what is the ideal food, what is the ideal fuel for the engine of cGAS-STING to trigger interferon. And I would go beyond the 8 gray. I would say probably when you treat photons, the breaks seem to be relatively consistent. But what if you go into protons or carbon ions? Are you going to now create different DNA fragments? I don't have an answer for it. Laura Santambrogio and I have been talking about a complex model of elution to look at the fragments with the different mechanisms, maybe in collaboration with Columbia University, where they have a RARAF machine that can mimic different LET radiation, but definitely a very appropriate question. And it may really end up explaining why work better than, they may be possible that particles work better. I don't have the answer yet.
Great. Another question from Tanya Gonzalez. As part of the immunogenicity induced by combination of radiotherapy and immune checkpoint inhibitors, is there any evidence of detrimental autoimmune side effects in these patients?
So I know as a personal communication that different groups have seemed to associate the occurrence, anecdotal occurrence of responses to radiation immunotherapy, particularly with anti-PD-1, with the occurrence of toxicities to anti-PD-1 or anti-PD-L1. We did not see it. We didn't see an increase in toxicity and more in field response or abscopal responses to radiation.
Thank you for that. We also have a question from Mohammed Zahid. In your conclusions, you identified a need to optimize usage of RT to induce these types of responses. What gaps in knowledge do you think need to be filled in order to facilitate this optimization?
So you will need many hours for covering this, so I'll try to be as brief as possible. I think what as radiation oncologists, because we are the main stakeholders of, is our tool that may make a difference in fighting cancer and maybe generating some cures. We really need to converge more, both preclinically and clinically. So I know many people are mounting trials. Some of the trials are outstanding, so don't get me wrong. And I'm learning from my own colleagues with some of the trial design being extraordinary. But you really need a coordinated effort, even preclinically, to do the experiments, to repeat the experiment in multiple labs, not to compete, but to synergize. And most importantly, you need to inspire a new generation of young radiation oncologists to embrace radiation immunotherapy. And that is a bit of a challenge, not that they don't exist, but you really need to create jobs and nurture them, find the funds to have them grow, and ask the questions we are not thinking about, and they are capable to think much better than us.
Okay, a couple more questions. Alice Ho says, thank you for that amazing talk, Dr. Fermenti. What biomarker do you think is the best one to show that RT has effectively vaccinated against the tumor when combined with immune checkpoint inhibitors? And I would add to this, is it possible to detect some of this double-stranded DNA potentially in the peripheral blood of these patients in order to maybe even fine-tune the remaining courses of radiation therapy that's going to be delivered?
So, well, hi, Alice, nice to hear from you. We are working very hard to try to develop a panel that of biomarkers that will give us early on in the course of the combinatorial, testing of radiotherapy and immune checkpoint, a hint of whether we're going in the right direction or not. And as I shown you, CD73 soluble seems to be a promising one. Certainly, when we look at type 1 interferon, we are testing in our patients. The presence or the absence of lymphopenia is important, but I think we need to go to a deeper level, and we really need to characterize, it's not just the number of lymphocytes, it's which lymphocytes they are. So there's a lot of flow cytometry studies to see whether are we killing memory T cells and or naive memory T cells. And also what I think is probably the most difficult challenge is to learn what the resident memory T cells are doing. And that, you can dissect in mice, but is much harder in patients. And last but not least, we really, and I take the entire blame, is my fault, and the people who did the abscopal studies over the years were justified because there was no science out there to show the heterogeneity of metastasis, the complexity of the crosstalk of metastasis. Now there is no justification. And there are so many beautiful papers in so many human settings, like the autopsy papers of Matos Deruda and so on, showing that, for the time you have in metastatic setting, you have really a coevolution of the immune system together with the genetic diversity of the metastasis. So if you want to immunize, you really have to take on all metastases and may not, so if you want to, it may be impossible to wait for multiple metastases just because then you induce lymphopenia. So you really want to work in the setting of oligometastatic disease and most probably do the least amount of radiation to be immunogenic, because also you want to keep the lymphocytes healthy and available for you.
You keep mentioning the lymphopenia, and I can understand why it's such a key. I'm wondering whether that leaves open the door to using flash therapy more effectively, with the higher dose rates, and therefore hopefully sparing more of the lymphocytes that may be passing through the fields.
Yes, we are just did the first set of experiments last week, and we are looking at the data. Yes, it can go both ways, right? So it could be extraordinarily immunogenic or may not. Certainly is a cytocidal tool. So for those who are not radiation oncologists, flash is the delivery of the same or more dose in an incredibly fast dose rate. So there are ways to tweak the radiation machines to deliver sky-high dose rate, thousands of times more than the standard dose rate. So instead of treating somebody over 2-3 minutes, you treat in a fraction of a second and give the same dose. And there is preliminary data that is at least as cytocidal, maybe even more, and protects the normal tissue, and that a complexity in itself. So is that protection in immune system, the normal tissue that Christopher is alluding to, going to play in mounting an immune response? Yes, very possible, and should be investigated. Yeah.
Well, we look forward to seeing some of that data in future publications and presentations. So we do have to end here. It's 5 past the hour, but thank you again, Dr. Fermenti, for joining us today and giving that fantastic talk. The remaining questions we'll have emailed to you so that you can also address them at your leisure. But thank you again, and we do look forward to seeing you again sometime soon.
Thank you.
Take care.